Okay, good afternoon, ladies and gentlemen, and esteemed guests and fellow students. I hope you are having a wonderful afternoon now, especially after our reception, the good food provided by our faculty. And I'm excited to welcome you to this William Mong Distinguished Lecture, which celebrates the remarkable achievement of Dr. William Mong, the founder of Sun Hing Group in the field of engineering and technology. He donated 10 million Hong Kong dollar in the year of 1991, quite a long time ago, to support the research and the knowledge exchange in Faculty of Engineering. And this William Mong Distinguished Lecture had provided a fantastic platform for the esteemed researchers from the whole world over the years to share the valuable insights with our professors, students, and also the broad community in Hong Kong. Today, we are thrilled to have Professor Wagner to join us to give this William Mong Distinguished Lecture on this topic. Seeing is understanding the next generation chemical imaging for super fast functional analysis of microbiomes. And to give you some background of Professor Wagner, this is the script prepared by the Dean's office for Dean, but unfortunately Dean cannot join due to some urgent arrangement. I need to go to US. So I will present that on behalf of the Dean. But I do not need this script anymore. And because for Professor Wagner, I know him for quite a long time. Actually, our first time is in the year of 2001, the second FISH course, talking about the FISH fluorescence in situ hybridization. I did learn a lot from that course and start my career as a molecular microbiology in this university. I think at that time I just a fresh PhD graduate and work at a postdoc in our department. I learned a lot. And after that, I pay much attention to the work done by Professor Wagner. And to give you a few examples, and in the year of 1994, they published a milestone work about the pores, the phosphate accumulating bacteria, organism in the wastewater treatment reactors, and correct the mistake already there for decades. But only with their work, they can tell us who is the real player in that reactor, using the fish and also other technology together to tell us it's Acuminobacter. It's a hero. It's a hero in that reactor to work for us. And also, I think another recent the groundbreaking work is on the Comomox. It's also milestone work on the nitrification. Tell us that single bacterium can do the oxidation from ammonia all the way to nitrate. Of course, still in 2 steps. The second step is from nitrite to nitrate, and the first step is ammonia to nitrite. So that also is a milestone work. And then, of course, there are many of these kind of milestone work. So Professor Wagner is very famous in this field, in our microbial ecology field. He was the president of International Society of Microbial Ecology in the year of 2014 to 2016, something like that. Yeah. And also he got Jim Tighe Lifetime Achievement Award from the International Society for Microbial Ecology. I think that's basically some brief introduction about the background, the contribution, achievement of Professor Wagner. For this lecture, I think Professor Wagner will tell us more. I believe it will be inspiring lectures to all of us. And before the lecture, on behalf of our faculty, we prepared a souvenir to Professor Wagner to show us appreciation for your visit and to share your wisdom wisdom with us. Yeah, please. I think page. Okay. Okay. Down. Okay. I should be taller. Okay. Yeah, they will take it and pack it for you. Yeah. Now, floor is yours. Please, please. Please.
Can you switch? Hello, everyone. Can you hear me well? Is this on? Yeah. So, Tong, many thanks for the kind introduction and for having me here. It's quite an honor to be here in Hong Kong. And actually, I would like to thank you for the invitation, for the honor, and also all of you who are here today and those who have made my stay so special also the last days. So many of you have accompanied me, hiking a little bit, going through for lunch and dinner and being on the ferry last night. And it was really quite an experience to see a lot in one day, Hong Kong in a day, basically. And it was really exciting. So thank you for making this effort and for having me here. I will have the pleasure to talk to you twice. So I have a talk today about the title which you see here. And there will also be a talk tomorrow at 11 o'clock on a totally different topic. These are my two main research interests I will present in these two talks. So today I talk about seeing is understanding. And interesting, yesterday at the ferry, I saw this Seeing the Unseen. That would have been maybe even the better title. So even at the harbor, they make a little advertisement for my talk. Seeing the Unseen by port. I think it's outdoor advertisements. So they know that this is important. So we want to see the unseen. This is what we are aiming for. Because we live on a planet of microbes, right? This is a planet. I start today very general. For those of you who might not be microbiologists, so the first slides are very general. Those who are microbiologists, don't get bored. There will be specific information later in the talk. But I would like to start. Why is this a microbial world? Because microbes dominate our planet, right? So we have an incredible amount of cells, bacterial and archaeal cells on our planet, 10 to the 30. You can't really imagine this. Many in the oceans, many in the soil. If you look at the biomass, this is sometimes we often forget. Plants, if you would weigh all plants, right, they are quite heavy. But then bacteria and archaea are contribute more to the earth biomass than all the animals, including humans, do. So if you put all humans together and all animals together, and animals here is mostly, cows and pigs, and so not natural, is wild animals, but those which we farm. And if you put them all together, they weigh much less than all the microbes on our planet. So I think this is, in a way, counterintuitive, all these tiny cells, and this tells you already how important they are for cycling of elements on our planet. And if it comes to species richness, so number of organisms, number of genes, bacteria and archaea, they outnumber all the other organisms. Look at the number of estimated species, bacteria, and these are animals, plants. Plants you almost hardly see, fungi and protists. But this is regarding diversity, this is a microbial planet, right? And it's also obvious to imagine because they were the first inhabitants of our planets. They had hundreds of millions of years, Earth alone, right? It could colonize all the niches and had a lot of time to diversify. So if you're hunting for new products, for new enzymes, bacteria will be the ones and archaea to look at. And the total amount of phosphorus and nitrogen in prokaryotes is 10 times higher than in plants. This is mostly carbon, the earth biomass. But if it comes to nitrogen and phosphorus, key nutrients, the amount is higher in microbes. And this is, as you know, this is the standard sentence. We consist of more bacteria than human cells. But 1 gram feces contains more than 100 billion microbial cells and several hundred microbial species. So in each of you, you have an astonishing amount of cells. And in total, there are trillions of microbial cells in and on the human body. And just to confront you with a few numbers, 1 gram of soil contains too many cells to imagine, and more than 100,000 microbial species. So if you go home tonight and you look at your flower pot, right, and 1 gram of soil there, you have 100,000 different microbial species in. So you don't need to go to a remote place to find new species, because after all these decades of hard work of microbiologists, only 15,000 bacterial and archaeal species are cultured. So if you contrast this, your flower pot in your living room, 1 gram soil, 100,000 species, all microbiologists in all different countries were able to culture only 15,000 bacterial and archaeal species. So there is so much uncultured microbial diversity out there, which we started to understand during the last decade, right? This was this revolution when microbiology started. When I began my PhD, I was lucky to have just the right timing. The molecular tools came up, right? We were all of a sudden able to sequence signature genes like the 16S ribosomal RNA, later on to sequence whole genomes, and to open this black box of the unseen majority of organisms on our planet. This was sort of the great revolution when all of a sudden one could at least tap into this pool of unknown organisms. And if you summarized the diversity of microbes of all organisms as we know of today, then you get this tree. This is a molecular tree. This is not fantasy. This is calculated by comparison of genes. And I would like to highlight a few things. These are bacteria, and this is a phylum level tree. So what I need to tell you is a phylum is something like the vertebrates, right? So it's a huge group of organisms. So behind every line here, which you see here, there are thousands of genera and species. So this is a very high level tree. Every line is a phylum. Keep this in mind. It's difficult, it's easy to underestimate this. So this is every line is a phylum. Behind every line, there is a huge diversity. These are bacteria. These are archaea. These are eukaryotes. The eukaryotes emerged, as you know, from the archaea. And you might ask yourself, where are humans? Where are you? You are here, one line. And you're not very special because you are here together in one lineage with fungi and amoeba. So just to tell you how broad these groups are, right? So humans are so closely related to fungi and amoeba that they are still in one line in this diagram. And if you digest this, so these are all animals, all fungi, all amoeba, one line in this tree. And this is all the rest of the diversity on our planet. So this, I think, gives you an impression, with what we are confronted. Life, if it comes to diversity, is microbial. It's not eukaryotic. This is a picture from my PhD thesis. I took this at the end of my PhD thesis. And I still like it because it also reminds me why I did this kind of research I'm currently doing. At that time, we were using fish. Tong mentioned this. Fluorescence in situ hybridization probes to visualize bacteria and archaea without cultivation. And it was the first time you could do this. So we could label microbes in their natural environment. This is a wastewater treatment plant sample. And it was fascinating. You saw these are 7 different populations which we labeled simultaneously, 7 different colors. And you could see for the first time all of the 7 are uncultured members. And you could, for the first time, give a morphology to them, determine their abundance, right? See how abundant are they, who sits next to each other, and you can then, over a year or so, do some, how do they fluctuate, how do they respond to environmental changes. But on the other hand, it was super frustrating at that time, 1990. The picture is from 1995. It was published in 1996. It was super frustrating because all we knew about these organisms is how they look like, how abundant they are, how they change in abundance over time. We had no clue. We knew only one gene of these organisms, the 16S ribosomal RNA gene. And that was the end of the story, right? You can make some correlations to changes in environmental conditions, but that was it. Super exciting to see all these new life forms, but also frustrating. So at that time I decided, and I stick to this throughout my career, to develop methods and apply methods and to understand the function of microbes. Not only to count them, like a stamp collection, but to understand them. And so we developed together with others. These are papers on purpose, not from our team. We were among the first to do metagenomic sequencing. Had a paper in 2008 where we published a whole genome of an Anamox organism. But by now, it's fast forward now, we can, with the new sequencing technologies, and you all know this, we can assemble whole genomes of uncultured organisms, right? These are just two relatively recent papers, and it's quite impressive, I think, to see that you have 200,000 reference genomes from the human gut microbiome. 200,000. If I still remember, for this one genome of the uncultured Anamox organism, it took us several years and 5 million euro together with Genoscope to get this together. And now, every PhD student can get a genome of an organism in an afternoon. But still, this is not the answer to everything. What we can do now, basically we have the tree of life. And first we populated it with 16S ribosomal RNA genes. Now we can populate it with genomes, right? Now we have sort of the whole tree of life, and you have a genome for every organism you like, basically. We are not quite there yet, but in a few years, there will be a genome for every lineage, for every organism. By the way, I forgot one thing I want to show you, because it is sort of important. All these lineages which have this symbol here, do you see this? This DNA symbol. They have not, again, these are phyla, right? They have not a single cultured representative. So if you go through this tree, you see among the bacteria and archaea many of these symbols, right? So we still have many phyla, you see this, without a single, not one member in the vertebrates, not one member which has been cultured. It's quite impressive, I think. And so we can get genomes for all these organisms. But does this tell us a story? Is this really so informative? One major problem is, wait, I need there should also be a magnifying glass. Yeah, here. If you look here, so these genomes from the human gut, 200,000 genomes, they encode 170 million protein sequences. But 40% of those lack a functional annotation. So for 40% of the genes in those genomes, we have no clue, not any idea what they are doing. It's almost half of the genes in the human gut, which is so well characterized, right, compared to a deep sea sediment or something. And even there, half of the genes, no idea. And if you want to listen to my talk tomorrow, I can show you that even for those genes where we think we have an idea, by sequence comparison, we are often very wrong. So what I want to tell you with this message, what is the message here? Message is you get genomes and you get annotations, and they are okay. They provide hypotheses, but often they are wrong, the hypothesis, and often we know nothing. We just know genes. And to illustrate this a bit further, I want to show one data slide from the Nobel Prize winner Frances Arnold. I talked to her several times when I was at Caltech during a research stay, a sabbatical. And what she does is she does protein evolution. So she tries to evolve proteins artificially in the lab so that they do new things. And that's where she got the Nobel Prize from. How is this related to our work? She works with cytochrome P450 and, for example, of this bacillus strain. What does this cytochrome P450? It hydroxylates long-chain fatty acids. So this is the substrate, and it hydroxylates it at a subterminal position. If she evolves this and cytochrome P450 and just replaces 23 amino acids, which is 2% of the total amino acid sequence. And if you think about annotation, 2%, if you would find a gene which has 98% sequence homology, you would put it in the same gene family, right? Every annotation tool does this. Here, 2% sequence change, and all of a sudden, this enzyme does no longer hydroxylate long-chain fatty acids, but it now is respecialized and hydroxylates propane. Totally different substrate. And you might say, this might have now a poor activity, but no, the activity of this modified enzyme is comparable with other naturally occurring alkane monooxygenases which convert this substrate. So 23 amino acid, and you have a very effective, with high product formation rates, new enzyme. And this is practical demonstration. But if you look in 1965, when I was born, Zuckerkandl and Pauling already made this quote in a paper: there is no reason to expect that the extent of functional change in an amino acid is proportional to the number of amino acid substitutions in the chain. Many such substitutions may lead to little change, whereas at other times the replacement of a single amino acid residue, by another, may lead to radical functional change. And I do think we hate this in a way, right? Because we have all these metagenomic data, and what does it tell us? We are often not wrong, right? Often homology is telling us something. But you always have to be suspicious. You can run into really bad surprises. And tomorrow I can show you some bad surprises, which at the end led to a really nice paper. So what I do think is the main message of this intro part of my talk is we need way more biochemistry, right? We need more heterologous expression of genes of uncultured organisms, and to study it's slower. No one wants to do it. Not many people want to do it because it's slower, it takes time. But if we want to increase knowledge, we will not increase it by sequencing more and more and more and more only. Because you can sequence as long as you want. It will never tell you the function of those genes. We need to characterize them by using biochemistry, by doing more physiology on pure cultures. We need isolates of these organisms, lineages at least, and by studying the physiology of organisms, so their function in their natural ecosystem. And today I will focus on this last task, in situ ecophysiology. So what do I mean with this? We in my lab, one of our approaches is to look at the function of organisms in their natural system. Because bacteria can behave very different in isolation, even if you can culture them. Often you can't, but if you can, they make different things when they are alone than if they are together with their competitors and partners. So if we want to understand what they are doing, we need to study them in complex systems. And meta-transcriptomics or proteomics is not enough, because for many of the genes, as I showed you, we don't know what they encode. So even if you sequence them as a meta-transcriptome, you still don't know what they do. So what we do to complement those approaches is we do single-cell isotope probing. And what is the idea here? The idea is you eat what you are what you eat. So if you are interested in who's eating cellulose, just to pick an example, and you add labeled cellulose, the organism eating the cellulose will become labeled. And if you can then, after short incubation, determine the identity of the organisms by a fish, and simultaneously for each of these cells determine the isotope composition by different methods which we apply, then you know who has eaten the labeled substrate. In my case, who has eaten the cellulose, just as an example. So we use different methods for this, microautoradiography in the old days with radioactive isotopes, and nowadays nanosims and Raman spectroscopy. Today I will almost exclusively focus on Raman spectroscopy. This is a Raman spectrometer. We do have a spontaneous one. We have 2 of these machines and a few more advanced machines I show you later on. It measures a Raman spectrum of a single cell. And basically, what Raman spectroscopy does, it's a vibrational spectroscopy. It measures all the, so you shoot at the cell with a laser. They all have the same wavelength, all the photons, right? Because it's a laser. And then if they hit chemical bonds in your cell, some of the photons are shifted in wavelength because they transfer energy to these chemical bonds. And this energy transfer is dependent on the chemical nature of the bonds. So what a Raman spectrometer in simple terms does, it measures all those photons which have a shifted wavelength. And this gives you information on the chemical bonds in your sample. It's basically a chemical fingerprint. And a single cell you can measure in a few seconds. Sounds fast, but if you think about the diversity, it's not fast. If you have 100,000 different species in a soil sample at different abundances, and you need a few seconds per cell, it's hard to measure those which are not very abundant. But we started with this. So, and we combined it, this is years ago, when we worked together at Oxford with Wei Huang, we combined FISH and Raman spectroscopy, which is not so simple because fluorescence is the enemy of the Raman spectroscopist. So we needed to find ways to have dyes which are not excited by the Raman laser or to bleach the dye. There are different ways. We can discuss after the talk if you want to. But at the end, we managed to combine this. So what we could do is we could go to a sample and determine the Raman spectrum. That might not be so interesting, but interesting enough, if the cell is labeled with a stable isotope, you are what you eat, some peaks shift. Look at this phenylalanine peak here, or it's basically the aromatic ring breathing of the phenylalanine. It is at 1003 wave numbers, and it shifts to 967 wave numbers if the cell is labeled with C13. Can you see this shift? This is the labeled and the unlabeled spectrum in gray and in black, in labeled and unlabeled. So you can, it just takes a few seconds, you can say, is a cell labeled or is it not labeled? And you can even quantify the amount of incorporated isotope. And the challenge then was that for many substrates, labeled versions are quite expensive or even not available. Often you have to synthesize them on your own. If you're interested in non-standard substrates, then it's pretty hard to get them, to purchase them. And if you purchase them, they're often not pure. So many challenges, very expensive. We said, can we have a more general approach? And in 2015, David Berry was still a postdoc. He's now a professor at our university. In my team, we developed together this heavy water labeling. So what is the idea here? If you grow organisms in heavy water, they will incorporate the deuterium from the heavy water via NADPH, you have chemical exchange there, into biomass. So whenever they synthesize a new molecule, they will deuterate their molecule, right? So active cells will be deuterated. And now this is a general activity marker. So whenever a cell is active, it becomes deuterated. And you can now design very simple experiments. You add your substrate of interest, let's say the cellulose, but you don't need it labeled anymore. You just add heavy water and cellulose, and the control is heavy water only. And then you look for those microbes which become deuterated in the presence of heavy water, which are not deuterated in the absence of heavy water. So and the beauty is, another beauty, just luck actually, the carbon deuterium peak in the Raman spectrum is in a region which is called the silent region. You see here why it's called the silent region. These are spectra, Raman spectra, from different pure cultures. Archaea, bacteria, autotrophs, heterotrophs, grown in different amounts of heavy water. 0%, 30%, 50%, 100% color-coded. And you can easily see, if you add more heavy water, there is a stronger carbon deuterium peak in a region where you can very easily identify it, because this is the so-called fingerprint region, because there are many peaks, and here it's silent, and you easily see the labelings. You're very sensitive because there is no signal usually. So we have now a generally applicable activity marker, which is also very cheap, because heavy water is cheap. And you need no labeled substrates anymore. You just add it, and you add your substrate of interest. And we even developed this further into a sorter. This was done together with Roman Stocker. He was at MIT, now he's at ETH Zurich. And so we built this microfluidic device, where all the cells, after heavy water incubation, they go through this device. You pick randomly cells out of the flow. You measure their Raman spectrum. And if the cell is deuterated, you sort it for cultivation. Cells are not killed by this, or for sequencing. And so this is a sorter, but a sorter not by fluorescence or by size, but by activity, right? It's activity sorting. It's the first time that you can sort out cells which do a specific function. And we applied this in the meantime for several studies. It still has not a super high throughput. So we sort 500 to 900 cells per hour. It has quite a good sorting accuracy, but it's okay, but it's not super high, the throughput. So this works nicely, and we applied this in many, many applications, papers, and projects, this approach. But all these techniques here have a very limited throughput, which really is the main hurdle of this stable isotope, single-cell stable isotope probing. So we teamed up with Ching from the Boston University Photonics Center and his postdoc, Chow. And on our side, the project I will now present was mostly driven by a fantastic postdoc at that time, Fatima Pereira. She's now a lecturer at University of Southampton. And I will show you one method development, and then its application, a real science story at the end of my talk, basically. So throughput is the challenge. So what we developed or adapted, Ching has developed really beautiful Raman technology, stimulated Raman spectroscopy, which uses two lasers, focuses on a certain part of the spectrum only, has and I could go into detail, but I won't, because it's not a photonics audience here, and I'm also not a photonics hardcore expert. But what this instrument basically does, it allows you to record this carbon deuterium, carbon hydrogen region of the spectrum in real time, almost. So it's in video rate. So you can look through the instrument, and you see carbon deuterium like you would see fluorescence in milliseconds. And you don't get a spectrum. You get a spectrum, but you don't visualize a spectrum. You visualize the deuterium labeling in basically real time, video rate. And to show you how such images look like so if you mix E. coli, and we combined it with fish, I have to say this, by adding a 2-photon device to this stimulated Raman spectroscopy instrument. So we combined 2-photon fluorescence with stimulated Raman spectroscopy. And so here, just to illustrate it, E. coli, Bacteroides, not labeled, 2 fish probes, you see blue and red, right? Same if you add labeled E. coli, blue and red. And this here is not fluorescence. This is chemical imaging. So within a millisecond, you see all the labeled E. coli. I hope you can see them here, right? All these dots. In the control, nothing is labeled. So for a microbiologist, this is really cool because you do basically activity microscopy, right? You do an incubation, and then you put it under the microscope, you combine it with fish, and you see who's active immediately. I find this quite fascinating. And here, these are the quantitative data, unlabeled bacillus, Bacteroides, unlabeled E. coli. And here, the mixture of labeled and unlabeled. And then you see the E. coli is clearly labeled in the carbon deuterium region. So now, for the first time, we also get very reasonable statistics with Raman spectroscopy because we can measure several 10,000s of cells within a day. I now show you one application where this and other approaches were important. So we are also interested in the human gut microbiome, and it's important for you. You know this for your immune system, for your health, connected to hundreds of diseases, and you can modify your human microbiome, gut microbiome in many ways. How you're born, cesarean section or naturally, breastfeeding, diet, exercise, disease, aging, where you live. In Asia, you have a different gut microbiome because you have more healthy food than in the US, and it's also visible if you look at the people. And so they are quite, it has an impact on many aspects. But what we often do not recognize is that the drugs we have to take when we get older. I learned recently that among the 55-year-old people, there's only a very small percentage not on medication. So at least in the Western world, there's a lot of people have to take drugs every day. And not drugs against bacteria, right? I'm not talking, in the rest of my talk, I'm not talking antibiotics. I'm talking human-targeted drugs for all kinds of diseases. And they change a lot our gut microbiome, but that has been overlooked for a long time. And actually, for me, this paper, it's not by our team, it's by Lisa Meyer and colleagues in Nature in 2018. This really started this project. I read this paper, it's really true, and said, I want to do research in this field. It's so important, and it's really interesting. I will do a project on this. And what Lisa showed in this paper is she analyzed 835 human-targeted drugs. And it's in a way conceptually very simple. She incubated those drugs with pure cultures of human gut bacteria. So a lot of combinations, right? And the surprising finding was 24% of these drugs inhibited pure cultures of gut microbes. But these drugs were for the brain, for the liver, for whatever, right? But still, they had a strong impact on microbes. And interesting enough, here you see the fraction of the drugs with antimicrobial activity between 0% and 100%. Here, the drugs, what they target. And if you look at the nervous system, so drugs which target the nervous systems, antidepressants, psycholeptics, antipsychotics, many of them with very low p-values, many of them have an effect on gut microbiota. And more generally, drugs, what she showed, change our gut, have an effect on gut microbes. But gut microbes also change drugs, right? They modify them, they transform them. So you have a complex network. In some way, bacteria are inhibited or stimulated by drugs. On the other hand, drugs are also modified. And then this might very easily explain why some of you respond to a certain drug and others don't, because you have different gut microbiomes, right? And this is a factor which has a long time been overlooked. We always think if we see these differences, which are very well described between different people, why drugs work on one person and not on the other patient, we say it's genetics. But it's not always genetics. Genetics is important, but it's often also the gut microbiome. And the vision would be in 10 years, if you go to a hospital and you need a drug, that they do not only do blood work, but they also analyze your gut microbiome and can predict which drugs work better for you and which drugs don't work as well for you. And many of the side effects of drugs, if you think about it, are gastrointestinal, right? Almost on every drug, if you look at the side effects, you have diarrhea and whatever. So it's also an interesting connection. And I think there are many opportunities for personalized medicine and new antibacterials here. So what we did is we looked at 2 drugs from the list of Lisa Meyer. So from her original work, we picked 2 drugs, loxapine and entacapone. This is a drug for schizophrenia, and this is a drug for Parkinson's disease. Very widely prescribed in the US, more than 30 million tablets per year. And we picked them and said, we don't analyze pure cultures now, but we look in the complex system, human gut. So we want to go one step further. What happens there? So what Fatima did is she incubated 6 fecal samples from our team with heavy water and the drugs. And it's important, we have 2 drug concentrations, low and high. Low is the concentration of this 2018 Nature paper, just to make a comparison. And high is the actual therapeutic concentration in your gut. So it's not an unrealistic concentration, it's the concentration you find as a patient in your large intestine. And then you incubate for a very short period of time. With FACS, you determine the number of cells. With amplicon sequencing, the community structure. With long-read sequencing, the metagenomes. And with SRS, you determine the activity. And the idea was, let's add rich nutrients here so that most bacteria grow, and we want to see which bacteria get inhibited by the drugs, right? We try to grow most of them and then monitor which bacteria in the complex community get inhibited. So let's look at the results now. So here, these are total cell numbers by FACS. These are the different treatments. What you see is most drugs, the bacteria grow. And that's what we wanted, right? We wanted that the bacteria grow, and most drugs overall, if you count all cells, they just grow. They are not very much inhibited. One exception, entacapone, the Parkinson drug, at high concentration causes quite a reduction and then a delayed growth, and still a very significant difference in total cell numbers. But for all the other treatments, no significance in total cell numbers, not resolved to the specific species, just total cell numbers. If you look at the same data set with our new method, with the stimulated Raman spectroscopy in heavy water, right? We did the incubations in heavy water. So then, same treatment, same comparisons. You see after 6 hours, look at the significance with the stars, we see already significant differences to the no drug control. And after 24 hours, they are even more pronounced. So what does this tell you? Even if the total number of cells does not change yet, you see if you look at the activity, you see already an impact of the drugs on the cells with all the treatments, even at the low concentrations and particularly at the high concentrations. You might ask yourself, why didn't he not include in the SRS analysis the entacapone high, which had the strongest effect here, right, in the FACS analysis? It's not here, not listed here. And you will see in a minute, cliffhanger, you will see in a minute why we did not include it here. This is a slide I would tell my students, never show such a slide in a talk. No one can understand it, too much data. And that's why I show it. But I will make it very simple. So don't look at all the details. What is this? These are absolute numbers, taxa resolved. So we combined the FACS data. So we know how many cells in total we have with the amplicon data. Then you get absolute abundances, right? So this is, in logarithmic scale, number of cells. The warmer the color, the more cells. This is the start, the mixture of these fecal samples, always in triplicates. Everything is done in triplicates, as you can see. These are the different treatments, no drug, different concentrations of the two drugs after 6 hours, after 24 hours. What you can easily see, the medium did work. You know, it becomes warmer, right? The most bacteria basically did grow over time. First message. When we then look at differences in abundances, and details not important now, in the entacapone, this is the Parkinson drug, low concentration, 4% of the taxa were affected. Entacapone high, almost 30%. Loxapine low, 6%. Loxapine high, this is the schizophrenia drug, 12%. So I find this quite surprising how many of the taxa were affected with statistical significance by these human-targeted drugs. Even if in total numbers you don't see a big effect, but in specific numbers you do see effects after. And it's even more pronounced if you do these activity measurements, which are not on this slide. These are the metagenomes which we have for the 25 most abundant taxa in this sample. I come back to this slide in a second. If we do this comparison with our activity microscopy with SRS, we even see stronger responses for taxa. So we made FISH probes for 6 of these taxa, and this is activity microscopy. This is not fluorescence. It's, I think, quite cool. Here you have the transmission picture. Here you have the E. coli, in this case, fish probe. And these are fecal samples. This is carbon-hydrogen, right? Every cell has carbon-hydrogen. And this is carbon deuterium. The warmer the color, the more active the cells. So you get such images, activity images, in a few milliseconds. And if you look, this is log two-fold changes. You see for all these taxa, after 6 hours, you see for almost all of them quite significant reductions, right, in activity. And much more pronounced, if you look at the p-values, much more pronounced than by sequencing. So again, the same message. Our method, the SRS, is much more sensitive. You see changes in activity if you see no changes in abundance. And this is important for many applications if you start thinking about it. So let's get back to this slide, and let's just focus on entacapone now with the red arrows. The therapeutic concentration of entacapone. So you see many microbes. Remember, 30% are really affected by entacapone. And most of them don't like it. You see, there are many which get blue compared to the original sample or the no drug control. So they are inhibited. But there's one outlier. This E. coli becomes really growing, right? Do you see this? They get really warm here. Much higher cell numbers. While other important members of the gut microbiome, like Bacteroides and Phocaicula, they get really, really strongly inhibited. So why is that? Why does a drug inhibit, a human-targeted drug, inhibit some bacteria and stimulate the growth of others? And that's where most stories stop. But we want to get to the mechanism, right? We want to understand what's going on here. And you will see in a minute that we know the mechanism now. But there is still one aspect we only partially understand. And this is what I show you now, and then I show you the mechanism. Before I showed you, we did not include the entacapone high measurements in our SRS analysis. Why? Because we noticed that entacapone has a signal in SRS analysis, which we did not expect. So when we incubated our our gut microbiome with entacapone, we all of a sudden, every cell had a very high carbon, or many cells had very high carbon deuterium peaks. We said, why are they all so active? That doesn't fit to the abundance data. Then we left out the water, the heavy water, and still they had a strong CD signal. We said, how can they have a carbon deuterium signal if there is no deuterium around? And interesting enough, just by coincidence, entacapone has a signal, a photothermal signal, which is exactly in this region where we measure carbon deuterium. So what does this mean if so many cells have this signal, unspecific signal? It means the cells apparently take up entacapone, right? And indeed, we visualize then entacapone using Raman, SRS. This is gut microbes. And you see inside the gut microbes, they accumulate the human-targeted drug. So they take up the drug which is intended for the humans, they take it up in their cells. And you can do a control experiment, so you can use dead cells, you see little accumulation, sorption basically, and with live cells from your human gut, you see a lot of uptake. And many different species do this. I don't guide you through the details. We did fish probes, and we checked E. coli, streptococcus, blah, blah, blah. They all accumulate entacapone. But interesting enough, this does not predict how they do later on in the incubation. Some of them who accumulate really thrive, right? E. coli, for example, it pumps itself full of entacapone and it grows. I showed you it's growing. Others, like Bacteroides, they accumulate a lot of entacapone, but they die. So apparently, they can't avoid taking up entacapone, but they have very different outcomes from this. Why? I have some speculation, but we don't know yet. Now, what is the mechanism? I talked to a chemist. I showed him the structure of entacapone. Thomas Böttcher, a professor in our team. And we were thinking about weeks what could be the mechanism. He looked at it and said, Michi, this looks like siderophore. And so siderophores are compounds which are binding iron. And microbes release them to bind iron. And if you look at it, he's right? Enterobactine of E. coli, it's a siderophore. It has this catecholate group. This looks sort of similar here if you compare these structures. And we digged in the old pharmaceutical, had nothing to do with bacteria then, literature, and they predict that entacapone can bind iron. So we used nanosims to say, do cells, these are gut bacteria, just a mixture, and we measured the iron content, no isotopes, just the iron content of the cells after entacapone entacapone exposure. And you do see here, if you add entacapone at therapeutic concentration, you have much more iron in the cells, right, than without. So there seems to be something going on with iron. We explored this further, so we used one of those organisms in pure culture, Bacteroides theta, B theta, which is, and don't look at all the conditions, look just at one condition. If you grow it, pure culture, cell numbers here on the y-axis. If you grow it with entacapone, it doesn't grow, right? You see this, no growth. If you add, this is entacapone here. If you now add entacapone together with iron, it starts growing again. So you can rescue the inhibition or the growth of the organism, you can rescue it by adding iron. If you do the same thing and you measure the total cells in a human fecal sample, you can also rescue it. So we took fecal samples. If you add entacapone, I showed you this before, they are inhibited. If you add entacapone and iron, they grow like in the no drug control. And if you look this now taxa resolved, these are the 25 most abundant taxa again. This is log 2-fold changes. This is entacapone. You see a lot of inhibition, a lot of warm colors, negative log 2-fold changes. Only E. coli is growing, right? It has a positive log 2-fold change. And if you add iron, most of this inhibition is gone. There are only very few organisms which are still inhibited, even in the presence of iron by entacapone. But for most organisms, no inhibition anymore. So iron makes a huge difference. And then we isolated E. coli from these fecal samples. And we looked also at the genome, and it encodes siderophore, enterobactine. We knocked out the siderophore and said, okay, what happens if we kill the siderophore? No enterobactine anymore. Then it's, wait, I have it here. Then you have here no drug. The mutant is growing like the wild type. If you knock out the siderophore production or its receptor, then if you add entacapone and the E. coli has no siderophore anymore or no receptor, it grows much weaker, can you see this, than the wild type. And if you add synthetically produced enterobactine to the E. coli, you rescue the mutant for the synthesis, but not the receptor mutant, as expected. Because if you add enterobactine and you have no receptor, you cannot make use of the enterobactine. So what this shows is that it's really iron, and it's really the enterobactine which makes the difference. Why E. coli can thrive in the presence of this Parkinson drug. So the model is, if you have your gut community, there is enough iron for those to grow here. If you add entacapone, iron is complexed, chelated, and only those microbes which produce siderophores can take up iron and will thrive. But this is bad for the patients because many pathogens do produce siderophores. And indeed, we saw that after incubation with entacapone, the antibiotic resistance profile and the virulence index, how many virulence genes are in the metagenomes, does significantly increase. So if you take this drug, you increase the likelihood that you're colonized by pathobionts. So with this, I summarize what I showed you. I hope I could convince you that Raman-based single-cell isotope probing is a cool tool for functional analysis of complex microbial communities. You can combine it with FISH or with sorting. And with SRS, we have now a high-throughput tool to measure activities of microbes with single-cell resolution. We increased the throughput by 2 to 3 orders of magnitude. And when we applied this to 2 widely used drugs, we showed that entacapone, and there are many other catechol-containing drugs. Entacapone is not the only one which has this chemical bond. So we would postulate that they all cause iron deficiency in the large intestine. And it was the first time that it was shown that iron deprivation is the effect of some of the, not all, but of some of the human-targeted drugs on the microbiome. And so with entacapone, you select for pathogenic microbes. And we now do experiments where we try to supplement patients with iron to rescue and have less of these side effects. But it's not easy because the uptake of entacapone in the small intestine is inhibited by iron. So you need to provide the iron in a form which is released only in the large intestine, not in the small intestine, to combine both things. With this, just last slide, entacapone is not given alone. The main drug for Parkinson is levodopa, which needs to go to the brain, produces dopamine, and helps you when you're a Parkinson patient. Unfortunately, levodopa is degraded by enzymes in your body. Entacapone inhibits one of those enzymes. So it's often taken together with levodopa to inhibit this degradation by the human body. And there's another inhibitor, this carbidopa, which inhibits another enzyme. So patients take more than entacapone. They have combinations of drugs. So we are currently investigating how these drug combinations affect, in Parkinson patients, the human gut microbiome. This is one of my interests. If you're interested in nitrogen, I know I'm running out of time. If you're interested in nitrogen cycle microbiology, which is the other thing I'm very much interested in, I invite you to come tomorrow at 11 o'clock to my talk. I will talk about our nitrification work, where we study these organisms which are really important for the global nitrogen cycle in much detail. And I will pick one example, our recent paper here, and show you a very new physiology of some of these organisms. With this, thank you again for your attention, for your patience, staying almost 1 hour with me here in this lecture hall. I would like to thank our center members. We are now 300 microbiologists. When I started in Vienna, we were 3 people. Now we are almost 300. And we are now mostly supported by, no, this is the wrong slide, the wrong direction, by a cluster of excellence. So Austria has decided to focus its research money on a few fields. And the first clusters of excellence were these 5. It ranges from history to Euro-Asian transformations, from quantum science to microbiomes. And we are proud to be one of those 5 clusters of excellence, which are funded each by 35 million euros for 5 years. And we are 32 PIs doing our work in this cluster of excellence. And so we have many open positions. So if one of you is interested to come to beautiful Vienna, just send me an email or look at our website for job opportunities. With this, thank you for your attention, and I'm happy to take any questions you might have.